set of slides, we're going to talk about skeletal muscles and create some muscle types and force. Our objectives are to distinguish between the two muscle fibers and explain how force is regulated. And we're going to look at summation, recruitment, and fiber types. We're also going to appreciate metabolism as well as energy sources in muscle. So when we talk about skeletal muscle fibers, we're really talking about two classifications, which are the slow ones and the fast ones. So we have type 1, which are the slow oxidative ones, and we have type 2, which are the fast oxidative glycolytic ones, and type 2B, which are just fast glycolytic. So what makes them fast and what makes them slow? Let's keep on looking at their characteristics. Okay, so just to compare type 1 and type 2, specifically the fast glycolytic type, we see that type 1 is small while the other type is bigger. Type 1 has a lot of mitochondria and glycolytic ones have few mitochondria. Because of the presence of mitochondria in type 1, they use ATP as a source of energy through oxidative phosphorylation, while type 2 obtains their ATP through anaerobic respiration, which is the use of glycolysis. Back to type 1, the increased ATP yields them to be non-fatigable, and so they can be used for endurance exercises such as walking and posture. While type 2 easily fatigue because of the low ATP levels, but due to their size, they're fast to generate force. So you use them for exercises that require big force, such as weightlifting. The type 1 are slow to generate force, and in terms of how they look, because of the capillary supply to the small type 1 muscles, they are red in appearance, whilst the type 2 fast glycolytic don't have much myoglobin, and therefore they look whitish in color. The human muscle itself has a mix of both type 1 and type 2. Let's look at the middle component, which is type 1A. How does it look like, or what are the characteristics of type 1A? So the characteristics we're actually looking at are those of type 2A. So as earlier stated, type 2 has been divided to include an intermediate one, which has a mix of both type 1 and type 2. So type 2A is a fast response of contraction that is more of type 2B. They have a lot of mitochondria, more the type 1 character, and they use oxidative phosphorylation to obtain their ATP, type 1 character. They are fatigue resistant, again a type 1 character. They are used during very powerful endurance exercises, so it's more or less putting type 1 and type 2B together, and they're red in color with a lot of capillaries and myoglobin. Okay, so we have talked about ATP as an energy source. So this is obtained from metabolism of carbohydrates and lipids, whilst phosphocreatine, PC, in the mitochondria, we see that ATP transfers a phosphate to phosphocreatine when the muscle is at rest. So during exercise, the PC can be hydrolyzed to form ATP for muscle contraction. Another energy source is food breakdown itself. So we see that at rest, lipids are mostly broken down for energy, but during exercises, carbohydrates dominate and undergo glycolysis to produce ATP. So this is both in aerobic and anaerobic conditions. So the aerobic conditions include oxidative phosphorylation, where we get more ATP, whilst for anaerobic, as we saw in the earlier introduction of type 2, produces fewer ATP. So getting back to our biochemistry, just a reminder, we see that we can get ATP from various levels. One of them being oxidative phosphorylation. This is coming in from the electron transport chain and oxidative phosphorylation within the mitochondrion. And this is as a result of activities from both the Krebs cycle as well as glycolysis. From the Krebs cycle, we see that the electrons are carried via NADH and FADH2 in order for oxidative phosphorylation to occur, while from glycolysis we see electrons being carried through NADH. But even through the Krebs cycle itself, we have substrate level phosphorylation going on that can be a source of ATP. Similar one is noted to come through from glycolysis, as glucose is being changed to pyruvate in the cytosol. So this is just a summary of how ATP can come about, and you have gone into details of these in your biochemistry. So looking at each type of muscle fiber, so to speak, we see that type 1 and type 2A have very similar sources of ATP. At rest, glycolysis and some mitochondrial activity is the source, while during exercise we have creatine phosphate giving a power surge of about 10 seconds as we start, and then glycolysis activities gives us ATP for about 2 minutes, and then for hours on end we have mitochondrial ATP. But in type 2B, in as much as at rest we use glycolysis, we see that creatine phosphate also gives that power surge at the start, and glycolysis also gives us a 2-minute surge of ATP. But there's not enough mitochondria to sustain, and so we see that there's this characteristic of being fatigued because there's no sustenance, unlike what we see in type 1 and type 2A. So in order for us to appreciate the force or the tension that is coming up in totality, we see that summation, recruitment, and the type of fiber are very key to appreciating gradation of force. So in summation, we are talking about the brain sending action potentials or impulses at increased frequencies, whilst in recruitment, what we are really stressing on is that increased action potential is sent to more muscle fibers. And just the type of muscle fiber also will give different types of forces, as we have alluded to in discussing type 1 and type 2 muscle fibers. So what we mean by the summation of action potentials is that with more stimulation you have more force. So if you have an electrical impulse that is coming in, it will definitely result in contractile force. Now, the contractile force has 3 periods, which are the latent period, contraction period itself, and then relaxation. So the latent period is just this period after stimulation where there's no contraction that is being observed. And this is due to the fact that calcium is flowing out of the SR and cross bridges have to be formed before tension is built, whilst contraction itself is the actual pulling and sliding of the filaments. And then we have relaxation after that, when all this activity is brought to a pause. To an action potential is known as a twitch. So like I said, the red one on top here is showing an action potential, and then the blue one at the bottom is showing the contraction response. So what you have is when you're having a wave of depolarization and the calcium is being released into the cytosol, you have this delay or the latent period, so to speak. And then when the sliding starts, then you can have the tension build up itself or the contraction, which is followed by the relaxation phase. So this is just to put side by side what the activities are that is between an action potential and the actual contraction, which is also known as a twitch. So the times for these periods differ with muscle types, that is skeletal or cardiac, so on and so forth. So on this slide, we are showing you a similar situation, but in this case, we are saying that the stimulation will take place at 0. And then with time, this is what you're going to see in terms of tension. So the tension, which is usually not at 0, will build up. First of all, you have this latency or this latent period, and we've explained why that is happening. And then for most of it, you have the contraction phase. And then after that, you have a slow relaxation phase that will take place. Now, I just want us to appreciate what the difference is between a slow twitch and a fast twitch, because in our typing of muscle, we're talking about slow contraction, fast contraction. So in the slow twitch, we see that there is an increased period of contraction, okay, while in a fast twitch it's pretty short. So it requires more time in the slow twitch to reach a peak force. And then there's also this increased period of relaxation. So we have less twitches that happen in a given time. While in a fast twitch, we see less time is required to reach the peak, shorter time of relaxation, and you can have more twitches in a given time. So what we have here is a comparison of fast twitch and slow twitch. I'll talk about them just now. All right, so as we can see on the diagram A, from the point of stimulation, we see the latent period, and then we see that it takes quite a while for contraction to actually take place. Now we're comparing this to diagram B, and let's look at the extraocular muscle. We see that it takes a pretty short time for it to get to its maximum tension, and then in the period of relaxation is pretty much from all the way 30 to about 140, whilst the extraocular muscle here actually takes very short time to get back to its initial tension. And in B is actually giving us perspective of 3 different muscles and how they can differ in terms of buildup of tension and release of tension. Something unique about the twitch in the cardiac muscle. Now, in the action potential of cardiac muscle, we observe that we have this plateau phase. Now, because of this plateau phase, we see that cardiac fibers remain contracted longer than skeletal muscle fibers. So skeletal muscle fibers potentials last maybe about 1 millisecond, but the cardiac muscle will last so the cardiac muscle action potential will last for about 250 milliseconds. So we see that the level of contraction or the time of contraction is also compared to the skeletal muscle is also longer. Now, someone might be asking, why is it longer? Recall that for cardiac muscle, we have entry of calcium from the ECF. So there's still availability of calcium to ensure that tension is still being built by processes of the myosin and actin. So that's why contraction is still taking place. So let's just talk about the frequency of stimulation. Now, we say the first stimulation in a skeletal muscle will cause what is known as a twitch. Now, if there's a second stimulation that is applied before the muscle can completely relax, it will cement or give a stronger contraction. And the successive stimulations, if they are given at the same frequency, will give this unfused tetanus, which we'll look at in a bit. So this is just a series of contractions with increasing force. But if you do a rapid stimulation without relaxation, you produce this smooth, sustained contraction. This one is known as fused tetanus. Let's just have a look at these. So in the first one, we have a normal twitch where we have the latent period contraction phase and the relaxation. Then in the second diagram where there is summation, we have an action potential at D, and then just before relaxation is completed, another action potential. So you see that there's summation there. If another action potential is given, you see that the summation will keep on going and you have stronger tension. But if they're given at frequencies that are regular, you're going to have this tetanus, an incomplete tetanus type of arrangement. But if you increase, if they're rapid, if the action potentials are happening in this rapid manner, you have this complete tetanus where you have slight increase, but it's almost a plateau there and you don't see any relaxations that are happening. So there is a phenomenon which is known where when muscles are initially stimulated when caught, they exhibit this gradual increasing response until they have warmed up. So the phenomenon is due to the increasing efficiency of ion gates as they are repeatedly stimulated. And the trapeze can be differentiated from summation because the strength of stimulus remains the same in the trapeze, but increases in summation. So here you have stimulations that are going on and you see that tension is increasing, but you are keeping the stimulations of the same strength. So a series of maximum stimuli delivered to a muscle at frequencies below that of tetany can cause developed force with each contraction. Now the force in this case is noted to increase in stages, and this is because there's a gradual rise in calcium concentration in the cytosol as well, because the SR pumps are unable to recapture them in time between the two stimulations. So that, together with the first reasoning where we're saying that the efficiency of the ion gates is increasing with repeated stimulation, can give us an understanding of why we have this tension buildup with each stimulation, and then at a point we just have this plateau of tension gain. So this is known as the trapeze effect or the staircase effect. So out there it's been noted that tetany does not occur in cardiac muscle. Why? So let's look at the skeletal muscle against the cardiac muscle in both action potentials and contraction responses. So if you see the buildup here, again, takes a little time compared to the buildup of tension in the cardiac muscle, and this is because of the longer action potential. So the action potential, which lasts much longer than that of the skeletal muscle, you find that the cell is in this absolute refractory period where there is no further stimulation that can happen. And so, because no action potentials can be generated until the repolarization is virtually complete, you can't have this Strape type of, or this tetany type of characteristic. So again, it takes a little while for the action potential to get back, and so the refractory period finishes quicker. By the time you're going to a third of the activity of the action potential, you can actually stimulate the muscle of the skeletal muscle again. However, in the cardiac muscle, you have to wait for it to come back to this stage, which takes much longer or to this stage where repolarization is complete and is not in a refractory period. Then you can stimulate it again. But by the time that it's coming to a complete repolarization stop, you will see that the response, the contraction response, is also almost at a full relaxation stop. So you won't have such a tetany kind of characteristic in the cardiac muscle. Increase of tension is not only due to increased action potentials or stimulation, it can also be due to recruitment, which is bringing in more fibers than just one. So how does the CNS do so? So we see that there's activation of what is known as a motor unit rather than just a muscle fiber. So a motor unit is a motor neuron. So on my right, you have two motor units. So it's a motor neuron. Let's follow the blue one, and all the fibers that it is innervating. So you have one in pink and one in blue. And you see that the fibers or the fiber numbers themselves vary in numbers in the various units. So you could have fewer ones in one unit and more. But what should be noted is that the fewer ones have this fine, precise movement, whilst coarse movement have fibers that could go to about 600 muscle fibers in a unit. So characteristically, the neurons innervate only one type of muscle fiber, that is, it's either type 1, type 2A, or type 2B, and never a combination of the muscle types. And of course, as earlier alluded to, an increase in the number of muscle unit contracting will increase the contraction force of the particular muscle. So the more units that are stimulated, the more contraction force of the particular muscle. So recruitment is actually the stimulation of motor units and adding them so that there is more force that is produced. So first of all, the smaller and the weaker units are recruited first, and then we have larger ones and stronger ones being added so that there is maximal force. But you see that with maximal stimuli, the activation of the motor units is such that it's only for a short period. So for example, the X plus Y there will only last for a short period, and then we'll have relaxation going on.